So, congratulations to Alan and all the previous gold Acta Biomaterialia Award winners, and certainly thank you to so many people who have made this possible. I actually would like to share this award with everybody that I have been a part of, and that includes this society. So I'm not a very good picture, someone who keeps photos. But I did get some photos from my collection of past members of my lab group, and really I want to share this wholeheartedly with the members of my lab. As we all know, there is a special relationship between an advisor and your students, and it's really their hard work, their deep effort that leads to all of our accomplishments in the field as well as individual accomplishments. And even though some of these awards are given to individual people, it really deserves to go to a group of people. In fact, one of the things I'm learning as a department chair now in chemical engineering at Northeastern is the more I stay away from my lab group, the more productive they are. So I think I'll continue to do some administration and see what we can accomplish as we go forward. I also really want to thank my advisor. Christy mentioned it was Rena Bizios, who is in the audience somewhere, for first bringing me to the Society for Biomaterials in 1997. So not only is the title of this talk 2 decades of commercialization, but it's also 2 decades of me coming to Society for Biomaterials. And if it wasn't for some of those initial inspiration, and motivation to come to a Society for Biomaterials that Rena installed in me, I wouldn't have found, I think, this community so much as a family. It's easy for us to think what's going on in our personal lives, professional lives, but it's so nice to come back to the same society every year and see similar faces and enjoy camaraderie, through having children in my own case, through moving to 3 different universities, but I always have this constant support from this society, and it's something I value tremendously, and I look forward to more and more years coming to the Society for Biomaterials. So, again, I want to share this with everybody, which I guess means it'll end up being about $1 for me, which is fine. But I do want to focus this presentation on commercialization. I think there was a couple of presentations earlier in the conference that perhaps were a bit negative on nanomedicine, so I'm a very optimistic person, and I wanted to share some optimism concerning commercialization of nanomedicine and some things you may not realize that are happening very quickly in this field, especially in terms of FDA approval that I personally have been part of and other people have been part of. So of course I'm going to talk about some of these commercialization efforts, and I need to give a disclosure for that involvement. I also wanted to stem this presentation around taking risks. I think this is a perfect society, and the presentations we've already heard of leaders in our field who have taken considerable risks in their research, which we are now benefiting from. So every time we think about our own particular research or education or industry job, whatever, it's so important for us to take calculated risks to move this field forward. And I think one thing we don't talk enough about in this society and research in general is failure from taking calculated risks. And in fact, that is something that should be embraced, and maybe we should think of ways to embrace failure from taking calculated risks more in our field. But one educational component that I wanted to highlight of when we took a calculated risk about 10 years or so ago was publishing in a new format. And that format is open access. And I know there are criticisms of open access, of pay to publish. There are a lot of benefits of open access that we started some 10 years ago with this particular journal. And it has to do with increasing exposure of research to as many corners of this earth as possible. Open access enables anybody with a computer or now an iPhone to download for free any paper that is published. So I'm a firm believer that increases the impact of our work. It allows people to get motivated about science, whatever that science is, whatever that engineering is, and it brings more people to our communities. So I encourage everybody to think about ways that we can further increase exposure of science and technology to a growing population of people, especially I think in today's climate where we might need to pay a little bit more attention to that kind of increased globalization and increased global exposure. And I know Elsevier also is doing similar things with taking risks in publishing and how we can increase the impact of what we're doing and the motivating the next generation of students to explore our fields. We've also, of course, as everybody has, been taking calculated risks in how we deal with healthcare. Healthcare obviously is an issue that is core to this society, and I wanted to present a framework of how we've been trying to deal with nanomedicine, nanotechnology to accomplish several things that are listed on this slide. Most importantly, I think things that we still need to face in this field are getting away from treating every patient the same, having a more generalized medicine I don't think has really served very distinct problems that people have, or has not been able to address the different immune systems, portions of immune systems that we have among each other. And I also think we need to move medicine to be much more predictive rather than reactionary. We still live in a culture of healthcare where we are reacting to your healthcare problems rather than predict them so that they wouldn't occur in the first place. And clearly, a lot of us, at least in those two specific issues of treating every patient the same and reactionary versus predictive medicine, we think there might be a solution in nanomedicine and nanotechnology. And we've all been pretty, I would say, drawn towards nanotechnology, not only because of the size scale and how you can develop small particles or small molecules, many of those we've already heard about this morning, to have increased circulation time in your body, but we can now probe cells, we can enter tissues, we can go to different parts of the body that you just simply can't do with micron or larger particles. We've also been very interested in nanomedicine or nanotechnology, not only in terms of nanoparticles, but in another part of the field which is called nanotextured surfaces. And nanotextured surfaces really allow us to mimic the natural roughness, the natural topography of our tissues themselves. It also enables us to explore this field called quantum physics, which allows for very distinct surface energies to be created through small manipulations at the nanoscale. And of course, as we know, if you start playing around with surface energy, you can start altering what kind of proteins absorb to implant surfaces, you can alter the bioactivity of those proteins, and ultimately affect cell function. So there are many reasons, I think, to get excited about this field that I share with many of my colleagues to really look at nanomedicine to improve our healthcare system. So I wanted to divide this presentation really into two parts, and it kind of follows the chronological order of my career, where one of those parts deals with self-assembled chemistries. Self-assembled chemistries has become a key part to the field of nanomedicine, and really self-assembled chemistries, or supramolecular chemistry that I think everybody is familiar with, is designing materials such as one like this, in which you control and develop new chemistries that self-assemble in a controllable way in a particular solution. So through secondary bonding or other molecular interactions, you can get defined self-assembled properties which, to us, can mimic the natural structure of our proteins in the body, thus providing a better regenerative medicine material. So this is just a slide of the very beginning of my career when I was at Purdue, in which we started looking at developing what we call helical rosette nanotubes with Hisham Faniri. These helical rosette nanotubes are composed of DNA-based pairs. Of course, if you want to develop a self-assembled chemistry, you should look no further than DNA or other natural materials in your body that self-assemble in controllable ways. We created, using these DNA-based pairs, a molecule. You can see up in the right-hand corner, a molecule which has complementary electron donors and acceptors. So when you place it in an aqueous solution, you get hydrogen bonds to form, about 18 of them, which bring 6 pieces of that pie together into a controllable self-assembled molecule. These molecules are fascinating to us because not only do you have controllable self-assembly in the XY direction, but you have controllable self-assembly in the Z direction, so you can control the depth of these materials. They can be great drug delivery molecules, having a more hydrophobic interior and hydrophilic exterior to allow you to, again, probe the body at this nanoscale. You can also put different peptide functions, which we have done, or peptides, on the outer part of this molecule to allow you to tailor these molecules towards cartilage regeneration or orthopedic regeneration or anti-infection strategies or anti-inflammatory strategies. So it's really a platform molecule, which a lot of supramolecular chemistry is, that can be tailored for particular applications. So of course I have a orthopedic type background, and one of the first applications we wanted to look at was in orthopedics. These are injectable materials that are liquid at room temperature, and at body temperature they solidify into a gel. If you add components like nanohydroxyapatite or other nanoparticles, you can mimic the mechanical properties of either cancellous or cortical bone to allow for a nice injectable self-healing process rather than utilizing a bone pin, a metallic bone pin, or even an artificial joint. You can see on this slide that we were able to increase bone growth through such approaches, and in fact at the bottom there, At the bottom there, you can see an approach where we use the rosette nanotubes with subregions of bone morphogenic proteins. We've identified 14 amino acid sequences that we can covalently attach to these molecules to, again, improve bone growth. So we've also explored these for things like siRNA delivery or other drug molecule delivery, where, again, you can utilize the hydrophobic inner core of the molecule to help deliver these molecules inside of cells and throughout the body. We have had a collaboration when I was at Brown University with Eric Darling and some of the people in the Rhode Island hospital system to really look at the delivery of molecular beacons to help improve imaging of certain diseases. So this is just an example of an animal study, which was an osteoarthritic animal study in which we combined a particular molecular beacon that was being developed by Eric Darling with our rosette nanotube, and we saw improved detection of a disease. So certainly, this led us at this point in my career to really look at a problem that we still have, infection. And actually, sometimes in clinical meetings, this is like the dark elephant in the room, or the thing that nobody wants to talk about. And somehow in this field, we have to get beyond thinking of infection as dirty surgery or something that goes wrong in the surgery room. Bacteria will be present in a surgical room, and the reason is because you will be present or the patient will be present in the surgical room. Wherever we go, bacteria go. So at some point, in order to really address this problem, I'm a firm believer we have to have accurate statistics in infection. We have to admit that we have bacteria problems. And certainly, as this slide highlights, we have to admit the growing concern of multi-drug resistant bacteria. In fact, you may know the World Health Organization has only gone to the UN to talk about 2 health problems in the history of the United Nations. The first was HIV/AIDS in the 1980s, and the second was last year to talk about the problem of multi-drug resistant bacteria, which, as you know from following perhaps some popular news, or if you're in the bacteria field, you know that patients are now have multi-drug resistant bacteria in their bodies when they have never taken the antibiotics that led to those multi-drug resistant bacteria. So this is a critical problem that we have to get a hold of, and I'm so happy to see with every passing year in Society for Biomaterials more and more attention being paid to infection and bacteria. So we've been developing self-assembled chemistries also to treat multi-drug resistant bacteria. Slightly different molecules than what I just presented, where there are regions that can attach to specific regions on bacteria membranes. But on this slide, you can see MRSA, Staphylococcus that's resistant to methicillin, or multi-drug resistant E. coli. And what we've been able to do, and other people report this for different self-assembled chemistries as well, that these self-assembled molecules basically can attach to a membrane and pry the bacteria membrane open, allowing the internal components of the bacteria to come out and, of course, killing those bacteria. So we all need to be very creative in thinking about how to kill these organisms that our antibiotics no longer work towards. The second half of this presentation is, as much as I love self-assembled chemistry and thinking about new chemistries in the lab, there's a big hurdle. to using these, as we all know, and this gets me into translation and commercialization. The big hurdle, of course, is regulatory agencies, no matter where you are in the world, and for good reason. There are big hurdles to keep people from using certain new chemistries in the body. So around the time I was at Brown into now Northeastern, we really kept investigating new chemistries, but we also switched into going back to what I call old chemistries. And I think this is another important thing that we need to consider as a society. We don't necessarily have to come up with new chemistries to treat every single problem that exists today in the human body. I think there's a lot of things we can do with our old chemistries that we have a lot of wealth of information about, but maybe we need to tweak them. Maybe we need to look at the process in which those new chemistries are formulated, the new process in which hip implants are formulated, or vascular stents. And in doing so, could we allow technologies quicker to get to the clinical side and be commercialized? So I wanted to highlight some of those efforts and really, again, draw our attention back to bacteria and draw our attention back to some of the problems we have in infection. And you don't have to look any further than the importance of nanotechnology in reducing bacteria growth than a cicada or a locust or whatever insect is in your neck of the woods. If you look at the wing structure as just one example of a cicada, and you look under a high-power electron microscope, you can see very distinct nanoscale features. These are very ordered, they're very regular, and we know bacteria do not grow on wings or some of these insects. So there's been a lot of studies really looking at the mechanism of how some of these nanostructures keep bacteria from attaching, keep bacteria from growing, that we can learn from in terms of using nanotechnology or nanotexturing for medical devices. Some of this deals with bacteria membrane stiffness, not being able to contort to these nanoscale features, and some of this also deals with surface energy changes you get by using nanoscale features, the quantum physics part of nanotechnology, to control whether those bacteria attach or not. So we've been taking those lessons from nature, and we've been looking at quote-unquote old materials and modifying them, and working very closely with startup companies to get either additional claims from the FDA concerning reduced bacteria growth, or getting outright approval to do clinical studies with these materials. So real quick, here's one particular example that's being commercialized by NanoV. This was a spin-out company from when I was at Purdue University, and it deals with anodization. Those of you in the medical device industry know how important anodization is for pacifying orthopedic implants. If you simply change some of the protocol, some of the recipe of anodization, you can develop nanoscale features on things like bone pins or other kinds of chemistries and implants that are used in orthopedics. Those types of nanoscale features can be carefully controlled to give you the energy you need to absorb natural antibacterial proteins from your body. And we have a lot of them. We have a lot of natural proteins in our body that if you can simply get them to absorb to your surface, you can keep bacteria from growing. So there's a whole thought process behind this that I don't have time to go into in real science and engineering that goes into what should those size scale of nanofeatures be. But nonetheless, when you do this, and we completed some animal studies, we see significant reduction of infection in devices that are used for amputees, for example, or even internal devices in which bacteria may colonize and grow a biofilm. So this slide is just highlighting a study that I did with Rory Aaron at the VA hospital system in Providence, Rhode Island, in which we were very interested, of course, in amputees, one of the most challenging biomaterials problems we have, I think, in all of biomaterials. Clearly, when you have an osseointegrated amputee device like what is pictured here, with a screw that's sticking out of the animal, you have to be concerned about skin closure to keep bacteria from colonizing that screw and therefore colonizing the bone. You also have to be concerned about soft tissue ingrowth and bone ingrowth. So when we anodized some of those pins, we were able to see in the second and third row complete skin closure, whereas using today's conventional pin, the wound remained open, causing consistent infection and consistent inflammatory tissue concerns. Another example that we've been involved with is collaborating with a company called America, who commercializes silicon nitride for spinal implants as well as other applications. Their material, I call it on this slide, smooth silicon nitride. We were able to nano-roughen or create nanoscale features on the smooth silicon nitride without changing chemistry. So we're not introducing new chemistries to the body, but rather new topographies to the body. We then consulted the FDA on what kind of experiments should we do to look at infection rates. It was suggested, and we came to the conclusion, to inoculate these surfaces with bacteria and then insert them into the calvaria of rats, look after a series of time, what kind of bone growth occurred, what kind of infection set in. On this slide, bone growth is in purple or blue, and infection or the presence of bacteria, biofilm proteins, is in yellow. If you quickly look at the nano-smooth silicon nitride, you can see about 10% of that implant was covered with bacteria, and there was about 15% that was covered with bone, but the image that's directly above is simply creating those nanoscale features, eliminated bacteria completely after 3 months. So there was no signs of any bacteria on that implant after 3 months, and that left a lot more room for bone to grow, so 41% of that surface was covered with bone. And please keep in mind, we are not releasing an antibiotic in any of these studies. This is simply changing nanoscale features to a controllable size range to give us attractive properties to reduce bacteria attachment and growth. So we do not think that this approach is contributing to our growing multi-drug resistant bacteria problem. Electrophoretic deposition, same idea. So this is being commercialized by a company called Tiber Medical, in which we're coating titanium implants with different size hydroxyapatite particles. We compare that to conventionally plasma sprayed titanium implants, and we're seeing the same exact result. This is an example for Pseudomonas aeruginosa. So we're seeing this example, and others are reporting this as well, not just in our lab, for gram-positive and gram-negative bacteria. Whereas many times you develop an antibiotic for that specific family of bacteria that you're trying to inhibit. The last example I want to give you before leaving with hopefully an inspirational note is atomic layer deposition. We're seeing the same thing. So atomic layer deposition has been very popular in the computer industry, and we're collaborating with a company called Ultratech, who is located in Silicon Valley, where you can use atomic layer deposition to create nanoscale features as well. And you can control this. You can have good control over the size of these nanoscale features by altering things like the temperature or pressure that's used in these atomic layer deposition systems. But what we're seeing, again, is the same trend in every example that I've presented so far of significantly decreased E. coli colonization when you create the right size scale of nanofeatures. And this happens to be titanium that was coated on top of titanium. So same thing with PEEK. There's a lot of interest in the orthopedic world about improving properties of PEEK, polyether ether ketone, one of the #1 implanted spinal materials. You can do the same thing, define a controllable nanostructured feature to decrease bacteria growth. And I could go on with a lot more examples of other research in the field that involves shot peening, cast molding, severe plastic deformation, consolidated nanoparticles. And our community is really seeing much promise in decreasing bacteria growth by using any of these processes, as long as you control and create the right size scale of nanofeature. So let me leave with just where I hope the field is going. And this is a big challenge to us. But every example, you remember in the beginning of the presentation, I talked about how important I believe it is to go towards predictive medicine and stop reacting to problems people have, because in a sense, we're always chasing our tail when doing that. My kind of hope for the future of the biomaterials field is really to go towards sensors. I think in the body sensors are a huge, promising area of research that can provide in real-time data what is happening inside your body. If you figure out how to do this correctly, have that information sent to a handheld device, much like today's pacemaker technology, but in this sense, using it more as a sensor. And then being able to use those same materials to control events if they're not going down the path you want them to. So just as one quick example, this is a sensor that we're developing that is off of an orthopedic implant where we anodize this orthopedic implant to possess nanotubes that I showed you in the amputee study. We grow electrically active wires or nanotubes out of the surface. Those electrically active wires can measure the conductivity of a cell that attaches. Now, bacteria have slightly different conductivity or resistance than bone cells, and bone cells and bacteria have slightly different resistance and conductivity than inflammatory cells. So you can use this as a mode to determine what cell is attaching to this implant after insertion. We've also been able to collaborate with some of our electrical engineering friends on how do you use radio frequencies to get that information out the body. And then lastly, we've created a controllable polymer that can degrade under an applied voltage to release a drug whenever you want it to release, for example, if excessive inflammation or infection is setting in. So, these kinds of approaches to me allow us to predict problems before they become incredibly challenging clinical problems that would require an implant removal. So I hope there's more work along those lines. You may also know in the nano community there's a lot of work going on in developing synthetic immune nanoparticles, so materials that can aid your immune system. They can attach to bacteria such as E. coli pictured here, release a content, and then kill those particles. And these would be great materials that are being developed in the nanomedicine field to aid immune system problems that a patient might have. So, really, of course, I think we have a huge area to go in nanomedicine, but to me, there are promising clinical applications that are occurring. There are ways to get through FDA approval quickly by taking our existing materials, modifying them to have nanoscale features so that you have chemical equivalency, but changes in nanotopography that have real changes in how cells respond to a material. So that we don't always have to think about a decade or 2 decades out for some of these nanochemistries to be used in the body. Some of this is happening today as we speak. Funding acknowledgements, and certainly, again, I thank the people who nominated me for this award, the letter writers, Elsevier for having this award, and everybody that's been involved, Art and Christy. And again, I share this with the whole society, a society that I love coming to and that I consider my home. Thank you very much.